The offspring of sexually reproducing species receive 50% of their genetic material from each parent. So you are 50% one parent, 50% another parent in terms of your biology. But then why are siblings different than each other? Think about it. Each of you are half one parent, half the other. So shouldn't you be very similar? I think all of you that have siblings, brothers or sisters, would argue you are very different than them. But you're both half and half. So why is it the case that you're different? Take a shot at it in a quick rate. Well, hopefully you can answer that by the end of this lecture. So we've talked about asexual reproduction before. That's reproduction that produces identical offspring. That's mitosis. We have one cell with a specific set of information, and it makes two copies that are identical to the original parent. These are exact copies. They might as well be called clones of each other. The exact same genetic information and the exact same number of chromosomes are passed down. And there are a lot of different examples of how asexual reproduction can occur. We just looked at mitosis, but budding is another one. Some creatures, like this hydra and this yeast here, will just grow miniature versions of themselves off of their body. And once it matures, it plops off, and now it's an entirely new organism, but it's a clone. It's identical to the parent. So with the rest of us, when you get more complex and you look at multicellular organisms, we can't do the same thing. Let's imagine that we did mitosis, and that was our primary method of reproduction. So here's an egg. Let's say it has 46 chromosomes, because humans, we have 46. And here's a sperm with 46. That means the zygote, the fertilized egg, is going to have 92 chromosomes. That is not going to work. That's way too much DNA. If we did it this way, every generation would have more DNA than the last. If that 92 chromosome baby grew up and reproduced, then I'd add 46 to that 92, and every generation we just have way too much DNA. This is not going to work as a way of reproducing. We have 23 pairs, and there's a reason why they're in pairs. By having them in pairs, that's going to enable us to overcome this problem. For example, think for a second. You have 46 chromosomes, 23 pairs. How many chromosomes did you inherit from each individual parent? You inherited 23. One chromosome 1 from dad, one chromosome 1 from mom, and now you have 2 of each of those chromosome 1s. This can work because chromosomes are homologous. A reminder that homologous just means same information. Tetrads are homologous. I apologize for saying tetrads, getting ahead of myself with this lecture. You'll know what that means in a moment. Chromatids are homologous. They have the exact same information. So in one chromosome, I have identical chromatids. But chromosomes are also homologous. If I have 2 chromosomes here, these both carry the exact same information. So the one from dad carries the same information as the one from mom. We also want to remember ploidy when we look through meiosis. Remember that cells that are diploid have one copy of each chromosome, but that's not always the case. Sometimes a cell has half of that material and it's haploid. It only has one copy of each chromosome. You can also have triploidy, octoploidy. We're going to mess with strawberries in a little bit. They are octoploidy. They have 8 copies of each one. In mitosis, all the cells were diploid. With meiosis, it's going to be a little different. To make sperm and eggs, we start with a cell called a germ cell. It's a cell that can turn into any other cell. Kind of like a stem cell, a little different. We're going to need to cut the material in each one in half. If I'm going to make a fertilized egg that has 46, I'm going to need to take that germ cell and produce an egg that only has 23 chromosomes, because you're getting one from each parent. So there's one copy of each of the pairs. Same thing with the sperm. That's going to need to be reduced to 23. We call sex cells gametes. A gamete is just sperm or egg. The reason we give it a different word is because it has a different number of chromosomes. All of the cells in your body normally have 46, except for your gametes. Your sex cells only have 23. By having that material, when sperm combines with egg, it will give us 46 total chromosomes. Perfect. This way, our offspring has the exact same number of chromosomes as the parents. This means we're going to have to alternate stages during fertilization. We started with the cell that was diploid. But then the sex cells became haploid. They were each 23. Those haploid cells are then going to combine to then go back and make a cell that's diploid. So with humans, we start with 46. Our sex cells have 23. Those combine and make a cell that's 46. This is what we know as our reproductive cycle. This is just showing you that process. What's going to make meiosis different than mitosis is that halving of DNA. With mitosis, I went 46 to 46. With meiosis, we need to understand how we can go 46 to 23 and then back to 46. So just to do a quick comparison, with mitosis, with asexual reproduction, I had one diploid cell at 46 chromosomes that created two identical diploid cells. Meiosis is going to be a little different. Our first division, we're actually going to go from one diploid cell, so 146, to two cells that are haploid, that have half the material. Reminder that 2n represents twice the number, so the number is 23. 2 times 23 is 46. So here I had 2n, I had two of each chromosome. Now I have two cells that only have one, so that could be a sperm or egg. But where meiosis is super fun is it doesn't only happen once. Meiosis happens twice. We're going to divide that cell again. When we divide it a second time, notice that the number of chromosomes didn't change. My first division cut it in half and made it haploid. But my second division, they're still haploid. I'm just creating identicals of that first two. So here's an overview of meiosis. Hopefully this diagram looks pretty familiar to you. I've got my nucleus, it dissolves. I've got oh, that's a little different. They're dancing, they're separating. We're going to use the exact same names to understand the phases of meiosis that we used in mitosis. For example, that's interphase, prophase, metaphase, and anaphase. But you'll notice there's a number after each one. We call this prophase 1, metaphase 1, and anaphase 1. We have to do that because there's two divisions that happen in meiosis. So the first round, splitting from one cell to two, we label each phase with a 1. For the second round, we're going to label each phase with a 2. So here's prophase 2, metaphase 2, anaphase 2, and telophase 2. Now there are differences in meiosis 1, all of these 1 phases, compared to mitosis, and I'm going to explain them in a minute. But know that meiosis 2 is identical to mitosis. Chromosomes appear, they line up on the metaphase plate, they split apart into sister chromatids that move to opposite sides, then I form my cell in telophase. So this is already good. You already know this. I'm not going to spend much time on meiosis 2. The focus of my time is going to be on meiosis 1 because that is new. So instead of saying IPMAT like I did before to help you memorize mitosis, now we have IPMAT, PMAT. IPMAT, PMAT. Let's go through the steps. With meiosis 1, I have interphase, prophase 1, metaphase 1, anaphase 1, and telophase 1. And I'm going to go 2n, so diploid, to haploid, just n. For meiosis 2, I have prophase, metaphase, anaphase, and telophase, and it's just n to n. It's just like mitosis, we're just making copies with the same information. Notice though, I went interphase, prophase, metaphase, anaphase, telophase. I skipped interphase. There is no interphase 2. I go straight into prophase 2, metaphase 2, anaphase 2, and telophase 2. There is a reason for that, but interphase is skipped between meiosis 1 and meiosis 2. For our first division, we're going to need to separate chromosomes. In mitosis, we separated chromatids, but notice in this diagram, I have chromosomes lined up next to each other instead of lining on top of each other. That separation means that each side now gets a full chromosome instead of a chromatid like they did in mitosis. In meiosis 2, it's going to be just like mitosis. We have chromosomes lined up in the middle, and each side gets a chromatid. So here we go. I want you to remember that the entire point of meiosis 1, the entire point of everything I'm about to show you, is to separate chromosomes and make haploid cells. That's our goal here. How do we go from 46 to 23? So let's go through each phase. Our first phase is prophase 1. Prophase 1, our chromosomes are going to appear and the nucleus is going to dissolve just like before. But what's going to be different is instead of having free-floating chromosomes, now chromosomes are going to attach to each other in homologous pairs, and we call that a tetrad. So a tetrad is two homologous chromosomes attached to each other. They attach around the centromere. So these could be two chromosome 1s, they could be two chromosome 3s. They cannot be non-homologous. There are no non-homologous tetrad pairs. So a chromosome 1 can't pair to a 3, 6 can't pair to a 20. It's always 1 to 1 or 2 to 2 or 3 to 3. We still don't have any diversity. I started this by asking you, why are siblings different? Well, right now, I just have the exact same information. Things get interesting later in prophase 1. When the tetrads form at the centromere, they're attached in the middle, they actually will accidentally break into each other. And the arm of one chromosome will collide into the arm of another one, and they will all break off pieces of each other and trade places. Think of it this way. If you were to run up and give someone a high five, and you traded hands when you did a high five, you now have that other person's hand and they have yours. That's what these chromosomes are doing. When their arms collide into each other, parts of the DNA break off and reattach. So we started with one completely red chromosome and one completely blue chromosome in this tetrad. Now check this out. We have diversity. We have multiple combinations of information now. This red chromosome has some blue on it, and this blue chromosome has some red on it. I now have a new unique genetic combination that didn't exist before the tetrads crossed over. Here's another diagram showing you those steps. So they attach as a tetrad, they cross over, they collide into each other, and then they exchange DNA. This creates an entirely new, unique genetic combination that didn't exist before. This creates diversity, which is a huge benefit to species evolutionarily. Why is it a benefit? Well, take a moment. What do you think? What are the advantages of crossing over in sexual reproduction? I told you it creates diversity, but what's the advantage of that genetic diversity? Why would I want to have different genetic combinations? Hopefully you see why as this goes on. So that's prophase 1. Crossing over happens and creates diversity. Chromosomes attach into tetrads. For metaphase 1, we're still going to have things lining up in the middle. But in mitosis, it was chromosomes. Now tetrads line up in the middle. So you can see in this diagram, I have this tetrad here lined up and this tetrad right here lined up in the metaphase plate. So tetrads will line up in the middle. Here's another thing that's unique though. Look at these cells down here. Something's different with each one. Here I've got all the blue on the left, here I have all the red on the right, but whoa, here I have 2 blue, 1 red, 2 red, 1 blue. Here I have 1 blue, 2 red, 1 red, 2 blue. There's a different combination of colors in each of these theoretical cells. This is something called independent assortment. When the tetrads line up on the metaphase plate, it is random what order on the plate they line up on. For example, here, if I draw a line down the cell, this cell of all blue chromosomes will become its own haploid cell, and this one of all red. So now I have 2 combinations. That's diversity. I have one that's all red, one that's all blue. But it could have randomly happened that only 2 blue are on the left, 1 red on the left, and then 2 red on the right, 1 on the blue. Well, now that's 4 different cells that could have been made, simply based on where they randomly line up on the metaphase plate. And again, this could be a cell 5, cell 6, cell 7, cell 8. Each of these random alignments that we call independent assortment can cause a new form of diversity. And the name makes sense here. The chromosomes are assorting independently of each other. It is random what side of the plate they line up on. This creates tremendous diversity in terms of the DNA organisms get, but what makes even more is these have already crossed over. This diagram isn't showing you that, but during prophase 1, these chromosomes already collided. So each one has its own unique combination, and what cell they go into is also unique. It's diversity on diversity on diversity. This is making more and more possible combinations for the sperm or egg. So we get to anaphase 1. Notice with anaphase 1 compared to mitosis, an entire chromosome is moving to opposite sides. In mitosis, it was only 1 chromatid. So now this cell's going to have 2 chromosomes, and this cell's going to have 2. So how is this different? We have an entire chromosome moving. Why is this a big deal? Remember, our goal is to go from diploid to haploid, to go from 46 to 23. This example started with a cell that had 4 chromosomes in it. Because of anaphase 1, now I have 2 cells that each have 2 chromosomes in it. So by moving a chromosome instead of a chromatid, I have cut the amount of DNA in half. Forming tetrads and splitting them is what makes that reduction of DNA. Then we get to telophase. With telophase, now each cell has 1 chromosome. Perfect. That's exactly what we wanted. So with meiosis I, we had 1 diploid cell split into 2 haploids. Diversity was created twice. The first time I made diversity was with crossing over in prophase 1. The legs of the chromosomes in those tetrads collided with each other. Second time was with independent assortment. The way they lined up on the metaphase plate was completely random, creating more diversity. Awesome, so success. Now we need to look at meiosis II. I mentioned to you before there's a phase that we skip between meiosis I and meiosis II. We skip interphase, but why? And I want you to think about this. I started with 46, now I'm at 23. Why would I skip interphase before going through meiosis II? What would happen there I don't want to have happen? Well, if I did interphase, what happens in interphase? I have G1, cells get bigger, and then S, the DNA duplicates. That's the last thing I want. If S phase were to happen, this haploid cell of 23 would just be turned back into 46. Well, that completely goes against the whole point of meiosis. I want to cut my DNA in half. So that's why interphase is skipped. If interphase did happen, the DNA would go back from 23 to 46, and I just did mitosis. It wouldn't really matter. So we had to skip interphase and jump right into prophase 2. Meiosis II, we're just going to do what happened in mitosis. So I'm not going to really spend much time on that because we already talked about it. Prophase 1, nucleus dissolves, chromosomes appear. Metaphase 2, you're going to have the chromosomes line up in the middle instead of tetrads like in meiosis I. They split into chromatids, and now I have 4 cells. 2 haploid and now become 4 haploid. Why is mitosis copied here? Because we want our cells to stay haploid. We're making gametes. We need cells that have half the material so that the embryo has the exact amount of DNA individuals of that species normally have. Putting it all together, we use meiosis I to make sperm and egg that are only 23. We did it twice. That way we have more sperm and egg. Those combine, and we now have a zygote of 46. That cell then reproduces through mitosis, and now we have baby. There's a lot of value to sexual reproduction. It introduces genetic variety or diversity. Independent assortment did that, and crossing over did that. There's also randomness added in what sperm meets one egg. There are millions of sperm. There are hundreds of thousands of eggs in an ovary. Which one ovulates out and which sperm meets that egg is also completely random. So you're adding diversity on diversity on diversity. This provides an advantage in evolution. Here's why. Look at this population of beetles here. Let's say that they move into a habitat that has really, really dark soil and their main predator is a bird. Since they're diverse, this beetle will probably survive. Absolutely, the bird won't see it. This one might blend in and that one might blend in. These two are going to get gobbled up. That diversity made it so that the species survived in its new environment. Imagine a population of clones where they're all this color, and then they move into an environment with dark soil. They'll all get eaten. The absence of diversity caused them to have a demise as a species. This works with disease as well. All of us, because of how diverse we are, have natural immunities to certain disorders. If any of those disorders strike on a mass scale, there will always be some survivors because of the diversity. Yes, there's a dark side to the diversity. A lot of us are born with disorders or diseases. A lot of us are more vulnerable than others. But evolution has made it so that that diversity overall ensures the survival of a species more so than if you were homogeneously one type or another. This is why each combination of gametes from a parent is unique. Eggs from mom and sperm from dad have undergone meiosis. Each of these have crossovered and done independent assortment. And which one meets is random. This means that every possible combination is unique and very unlikely to occur again. If you were to do the math, any two parents will produce a zygote with over 70 trillion different possible combinations. 70 trillion. You are 1 out of 70 trillion possibilities of what your parents could have created. That's pretty exceptionally rare. But there's still a family resemblance. You can still tell when someone's related to someone else. Here's the Jonas brothers, here are the Baldwin brothers. You know they're related. It's because each person you're working with the same 50%. But what combinations of that 50% you get is randomized. So you're working with the same base materials. If I write a book in English, I'm working with 26 letters. But there's trillions of ways I can combine those letters to convey a different story. But I'm limited by those 26 letters. Let's review. So here's a diagram. What is the name for the structure that this is pointing at? That's a tetrad. So I have two homologous chromosomes attached together. Let's try another one. Each of these chromosomes are represented by two colors. So that's some blue with a little red, that's red with a little blue. What happened to them that caused that? Why are they no longer just red or just blue? Crossing over. Crossing over during prophase 1, those tetrads collided into each other and exchanged information. A diploid cell is going to undergo or do meiosis. After it does meiosis, how many cells are going to be produced? And what is the ploidy of those cells? We're going to make 4 cells that are haploid or with half the material. So mitosis is 2n to 2n, meiosis is 2n, and 4n total. Last question. This diagram shows during metaphase 1, each cell will receive one mostly red and mostly blue chromosome, right? This will receive this, and this will receive this. But it could have been any other way. It could have been 2 blues here and 2 reds here. What do we call that random alignment along the metaphase plate? It's independent assortment. It's one of the 2 main causes of diversity in gametes. Just so you know, the reproductive cycle is very different across the animal and plant kingdom. This diploid to haploid to diploid, a lot of mammals do that, but you have algae, frogs, plants, they can go in completely different directions. There are some plants that actually get more DNA every generation, something called autopolyploidy and allopolyploidy. Very, very diverse and rich variety of how living things produce genetic diversity throughout the kingdoms on Earth. So let's do a quick comparison of mitosis and meiosis. With mitosis, I had 1 division. With meiosis, I had 2. Mitosis, I had identical offspring. Meiosis, every cell made is unique from the parent cell because of crossing over and independent assortment. Mitosis made 2 cells. Meiosis made 4. Mitosis, I went 2n and 2n. Meiosis, I went 2n to 1n. Mitosis is for making clones, so it's for growth and repair, whereas meiosis is strictly for gametes, for sex cells and reproduction. There's no crossing over in mitosis, so there's no diversity, but in meiosis there absolutely is, creating that diversity. So let's talk a little bit about things go wrong. This is a real case study, by the way. This is something that affects a lot of people. During metaphase 1, spindle fibers normally attach to each side of the tetrad so that each side gets 1 chromosome. But check out what's happening in this diagram. Here, there was no spindle fiber on the right. So now I have a tetrad moving this way and nothing over here. What effect do you think that's going to have in the 4 cells that I end up producing at the end of meiosis? Make a prediction. Well, since this cell's getting more DNA and this cell's getting less, I actually end up with 2 gametes that have 1 too many chromosomes, chromatids pictured here, but they reproduce later, and 2 that have 1 too little DNA. Think about the ramifications of that. If you have a fertilized egg, that means every cell in that person's body is short 1 entire chromosome or has 1 chromosome too many. These chromosomal abnormalities are somewhat common. One of the most common ones is what I just showed you, which is called non-disjunction. Disjunction means to separate, so non-disjunction, a failure in separating. That's when they don't separate properly, but what else can happen is chromosomes can break. A part of them could be deleted or unnecessarily replicated, or the recipe can be reversed. So let's go back to what I showed you before with non-disjunction. If that separation happens incorrectly, I'm now going to have gamete with one too few, gamete with one too many. This causes an alteration in the total number of chromosomes an organism has and creates what we call a trisomy. A trisomy is when a cell has 3 copies of a chromosome instead of 2. You can also have a monosomy. That cell that had one too few now only has 1 copy. And those can cause a variety of different disorders. Pretty high frequency in humans. For example, Down syndrome. Down syndrome is commonly referred to scientifically as trisomy 21 because in the 21st pair, there's a trisomy. There's 3 chromosomes instead of 2. That additional copy happens in about 1 out of 700 children born in the US. That is fairly common. We have hundreds of millions of people in the United States, so 1 out of 700 is frequent. And chromosome 21 is the smallest chromosome, but it can still have a large variety of symptoms in your development from that extra DNA.